Hi everyone. We're going to be picking up where we left off on Mendel's laws on patterns of inheritance. So just to recap what we talked about last lecture, Mendel did monohybrid crosses, which is when he kind of just examined one character trait, and he noticed that the big observation that he made was that dominant alleles tend to mask the recessive alleles. So when you have a dominant allele and a recessive allele together forming a heterozygous organism, you will only see the phenotype of the dominant allele. So he wanted to understand how traits were inherited, whether or not when one trait is inherited, if it affects the way that other traits are inherited. And so he did something called a dihybrid cross. So instead of just looking at one trait like in the monohybrid cross, in a dihybrid cross he looked at two traits. So in his experiment, he started with the P1 generation, which just means the parent generation, where he had two homozygous organisms, and they're both homozygous in different ways for both traits. So one of the parents was homozygous dominant for both its color and its shape. And then the other one was homozygous recessive for both of its for both the color and the shape. So remember, dominant is yellow and smooth, and then the recessive is green and wrinkled. So these are just terms that you should know. Dominant here, recessive, and both of these are homozygous because they have the same alleles. So he took that and he crossed them together, and he got this as its offspring, the F1 generation, which is just the offspring of the P generation. And all of the offspring were this. They were all heterozygous, and because they were heterozygous, you only saw the phenotype of the dominant allele, so it was also yellow and smooth. So Mendel took the offspring for the F1 generation and fertilized it with itself, so you had two heterozygous peas that were mated, and he wanted to know what the offspring would be for the F2 generation. And he was trying to determine whether or not these two separate characters were assorted dependently, where the genes are inherited together. If you have one gene for one of the traits, for one of the characters, you'd have to have it would affect the genes of the other trait that was inherited. Or it's possible that the genes were inherited independently through independent assortment, which means that the genes that are inherited from one trait, it doesn't affect the genes that are inherited of the other trait. Now remember, due to the law of segregation, or in other words, the regular process of meiosis, each parent cell will only donate half of its genetic material to its offspring. And so in the formation of sex cells, these heterozygous parents will only donate half of its material. Now, under the assumption of dependent assortment, this would mean that if one of the traits was dominant, the other trait would have to be dominant too. So in this case, it would be dominant color, dominant shape. It would have to be yellow and smooth. So this would be one of the sex cells, or they could both be recessive. So if one of the traits was recessive, the other one would definitely have to be recessive. So you see how one trait is affecting the other trait. So to determine the offspring, to cross this, we would create a Punnett square. And remember, each one of these heterozygous parents can only produce sex cells that are either both dominant or both recessive. And when you cross this, the sperm cells and the egg cells together, these are the only combinations you can get for offspring. So each one of these little small boxes represents the offspring, and you can see the only possibilities are smooth and yellow or wrinkled and green. Under the case of independent assortment, each of these traits, the trait of the shape and the trait of the color, are inherited independently. So you could have definitely both be dominant and both be recessive, but you could also have one be dominant and one be recessive, or vice versa. So dominant color, dominant shape and recessive color, or you could have recessive color, and dominant shape. So these are the 4 different possibilities of the sex cells for the parent that need to be crossed with each other. So here's the different possibilities. You have the sperm cell from one heterozygous parent could be big R, big Y, could be big R, little r, big Y, little r, little y. And same goes for the egg cells. It could be any of these combinations. And so when you cross all the different possibilities of the sex cells of both of the parents together, you have 16 of these different combinations. Notice that some of them are repeats, but you have a lot more possibilities for the offspring. And so when Mendel did this experiment, he found that he did not find dependent assortment outcomes, but he actually found his results supported independent assortment. And so this is the way that genes are inherited. They're all inherited independently from one another. So just as a recap, he had the P generation of both being pure true breeds. Remember, each of these, this parent here can only donate RY. That's the only combination. This one can only donate little r, little y. And so when you cross these two, the only possible offspring is a heterozygous offspring. So your F1 generation is heterozygous. And so then he crossed 2 heterozygous offsprings in the F1 generation. And remember, each of these parents can produce 4 possible different combinations for its sex cells. And so when you cross these different combinations for the parent sex cells, you get the F2 generation. The F2 generation can be any one of these little squares. So any one of these little squares represents a possible offspring for F2 generation. So what he found in summary was there was about 10 distinct genotype possibilities, but 4 distinct phenotypes. So in terms of the 4 distinct phenotypes, they came in a 9:3:1 ratio. This ratio here is really important. I would write this down in your notes. 9, remember, phenotype is just the physical characteristics. So he saw 9 out of the 16 different possibilities were dominant. 3 were dominant recessive, so one of the traits was dominant, the other one was recessive. 3 were recessive dominant, so that means that the other trait was recessive, but the other trait was dominant. And then 1 of the 16 would be recessive. Okay, so switching topics, we can use these principles of Law of Independent Assortment and the Law of Segregation to do a Testcross. A Testcross is something that we do to determine an unknown genotype of some kind of organism. So we take them, we mate an unknown genotype, an individual of unknown genotype, with a homozygous recessive individual. The reason why we do this is because we want to determine the genotype, and based on the offspring of this Testcross, we can figure out what the genetic makeup is of this unknown organism. So there's 2 different possibilities when you have an unknown genotype. If it's like expressing a dominant allele, it could either be dominant homozygous or dominant heterozygous. If you cross it with a homozygous recessive individual, you can have these 2 as possibilities. If the original organism happened to be homozygous dominant, then all of the offspring in the cross of these 2 would be, all of the offspring would express the dominant allele. Whereas if this right here, the original organism, was heterozygous dominant, then the offspring could either be dominant, or it could also possibly express the recessive phenotype. And so based on the offspring here, you can figure out what the original genotype is of this parent organism. So there's some probabilities involved in genetics, and you can use this to kind of save time rather than making a huge Punnett square, especially if we're doing multiple characteristics. So if we consider like just tossing coins, the probability of getting heads is 1/2 or 50%. And the outcome of the coin toss is unaffected by the previous attempts, which means that if you flip heads and you want to predict what the outcome will be when you flip the coin again, it doesn't matter that you just got heads. It's still a 50-50% chance that you will get heads. So there's something called the rule of multiplication, and this calculates the probability of multiple separate events happening in a very specific sequence. So if I wanted to predict getting 3 heads in a row, you would multiply each possibility of getting heads, so which is 50%, and multiply it 3 times. So 50% for the possibility for 1 heads, multiply by 50%, multiply by 50%. So you have 1 in 8 chances that you'll get 3 heads in a row. So this applies to genetics when you want to predict like a specific offspring. So let's say you cross 2 heterozygous parents, and you want to predict if you can get homozygous dominant offspring. The chance that you would get one big B or just the dominant allele for one of the parents is 50%. It's just 50-50. And the same goes for the other one. The chance that you're going to get the dominant allele for this parent would be 50%. So if you multiply those two 50%, you have 1 in 4 chances of getting a homozygous dominant offspring. You can also easily figure this out by doing a Punnett square, but the reason why we're doing this in a mathematical way is sometimes the Punnett squares get too cumbersome. There's also something called the rule of addition. And so this predicts the probability that an event can occur in multiple ways, and you can determine that by adding the probabilities of the different alternatives. So what I mean by that is, let's say you're crossing 2 heterozygous parents, and you want to predict what's the possibility that I'll get any kind of heterozygous offspring. You have a 1/4 chance that it will be big B, little b offspring. Remember, 50% chance that you get big B here, 50% chance that you'll get little b from this parent, and so 1/4 chance overall. But you also have to account for the fact that you could get little b, big B, and it's the same 1/2 times 1/2. You get 1/4 probability, and so both of these are possible outcomes that could tell you whether or not both of these are heterozygous. So if you add up these 2 probabilities, the probability of getting this possibility and the probability of getting this possibility, you get the overall probability of just getting a generic heterozygous offspring. Okay, the last section talks about pedigrees. Pedigrees are like a family tree that describes the traits of parents and children over many generations. And so it looks something like this. There's a couple tips that you can use to interpret both the genotype and the phenotype of like the different generations. So females are usually circles, males will be squares. If you can always tell the genotype of a recessive phenotype. So if you see that one of the family members is recessive, has a recessive phenotype, automatically you know that its genotype has to be recessive. Dominant phenotypes is more difficult to understand because it could either be dominant homozygous or dominant heterozygous. And so to determine which one is which, you have to look at the offspring of that family member. And so just keep in mind what we talked about with Testcrosses. Based on the offspring, you can sometimes figure out what the genotype was of the parent of the unknown organism. All right, thank you so much for watching. Bye.